Hello friends, in this video I will be talking about hybridoma technology. Hybridoma technology basically it's a biotechnological process in which enormous amounts of antibodies are produced using the B cells and the myeloma cells. So what are basically antibodies? Antibodies are immunoglobulin molecules which can specifically target a pathogen or any molecule. So it can neutralize a pathogen by binding with its receptor or anything. So it can also help in opsonization process so that the immune cells can easily detect the pathogen, or they can also activate the complement system. So antibodies can be of two types. One is polyclonal antibody, and the second one is monoclonal antibody. Polyclonal antibody means that the antibodies are produced from different B cell clones, or it's a mixture of antibodies. But the monoclonal antibodies, these are a specific type of antibody which are produced from a single B cell clone. So all the antibodies are actually same. They have the same epitopes. Hybridoma technology basically it's a method of forming hybrid cell lines called hybridomas by fusing a specific antibody producing B cell with a myeloma cell, which is a cancerous B cell. So and it can produce enormous amounts of antibodies. So Professor Caesar Milstein and Professor George J.F. Kohler, they first discovered this technique. They invented this technique, and they shared the Nobel Prize also for the hybridoma technology. So in hybridoma technology, B cells, we know that the B cells are antibody producing cells, but they don't have the immortal property, so they will die after a few time. But the myeloma cells, they have the cancerous property, so they can grow enormously. So after the fusion, they will produce a type of cell that is called hybridoma cells. These are the cells which are the fusion of B cells and myeloma cells, and they can produce enormous amounts of monoclonal antibodies. So they will produce large amounts of antibodies. So that is the basic idea of hybridoma technology. So for the hybridoma technology, first we need to immunize a mice or a rabbit with a specific antigen to stimulate the antibody production. Second one, then we need to isolate the B cells from the mice or rabbit for a particular B cell. Then we need to fuse it with the myeloma cells. After the fusion, it will produce different types of cells like the fused or unfused B cells, or the fused or unfused myeloma cells, or it can produce a large amount of hybridoma cells also, which are the cells the fusion of B cells and myeloma cells. Then we need to select which are the hybridoma cells by using the hat media and ELISA also. Now we have to expand it in a selected the selected hybridoma to produce large amount of antibodies in media. To do this, we need the media, specific media, that is called hat media. Hat means hypoxanthine aminopterin thymidine medium. So hypoxanthine, it's a precursor in the synthesis of DNA. Aminopterin, basically it's a folic acid antagonist that inhibits the de novo synthesis of the nucleotide synthesis pathway by blocking the activity of the enzyme dihydrofolate reductase. Thymidine, it's a nucleoside that can bypass the blockage caused by the aminopterin. So DNA synthesis pathway can be done by two biosynthetic pathways. One is de novo pathway, another is the salvage pathway or the alternative pathway. In case of de novo pathway, dihydrofolate is converted into tetrahydrofolate by the help of enzyme dihydrofolate reductase. But if aminopterin is present in the medium, it can actually inhibit the production of tetrahydrofolate. In case of salvage pathway, it uses AGPRT enzyme. It basically uses defined precursors like the hypoxanthine and thymidine, which can be converted into nucleotides, any nucleotide. So in case of hybridoma technology, we need the myeloma cells which are AGPRT negative. AGPRT negative means they cannot produce AGPRT enzyme. So the salvage pathway will be gone for myeloma cells. They cannot produce the nucleotides by the salvage pathway. So it can be done while myeloma cells are basically treated with 8-azaguanine. Most of the cells will die, but some of the cells will be viable, which are AGPRT negative. So after the fusion, three types of cells we will got fused B cells, hybridoma cells, fused myeloma cells. In case of fused B cells, these will die because they are not immortal. After a few weeks, they will die eventually. Hybridoma cells will grow rapidly and they produce enormous amounts of antibodies. And fused myeloma cells, they will die first because they are AGPRT negative, and also the de novo synthesis pathway is blocked by the aminopterin. So that's why they will die first. So now we need to grow the hybridoma cells in specific medium to produce large amounts of antibodies. So please like, share and subscribe to my YouTube channel. Thank you.